Hi everyone, come back after the mutation section. It's about the RNA-seq. RNA is just like we measure the gene production level, but instead of measure from the final product protein, it's quite hard because the structures and the current technology does not support it well. We can measure it via the intermediate product called the mRNA, or for example the protein coding gene. And right here what we need to do, just like we extract the RNA and then we can capture them and then we can convert into the cDNA right here. The reason is just because we already have protocol to sequence for the DNA. The second thing is about that the mRNA is the out of the RNA is not stable, just a single strand, and as a result we need to convert into the cDNA, and later we can just like do a normal DNA sequencing, but actually we can align into reference genome and we can get back the idea of how a gene that is expressed, which means like we have more read that can align into the region of gene, which means they have more expression level. And why we need to do the RNA sequencing is just like we will sequencing not a single sample but a lot of sample, and from that we can compare the gene expression level between them. As a result we can understand that what's going to be the gene that are differential expressed between group. For example, we compare the cancer versus normal group patient, and then we can understand like maybe there's going to be the gene that's overexpressed or less expressed between the group. So we can focus more on that to study. And actually for RNA-seq data we can sequencing for more than 20,000 encoding gene at a time, instead of for the technique like PCR, real-time PCR, to measure just like a single or a few gene at the same time. And to do that we need to take care a little bit about how we compare between group of sample. So I will use two example right here, just like sample A represent for a group of, for example, cancer, and sample B for a group of sample, for example the normal one. And then the first topic is about if we want to compare, the first issue we will encounter is about the gene expression, the total depth. For example, we have the sample A right here. All of the gene they have a higher depth when we compare with the sample B, which means like it will be imbalance when we compare between sample A and sample B, which means like nearly all of the sample A will have a higher expression level than the sample B. The second issue of the how to measure the gene production is about the length of the gene. For example, we have gene X and gene A. So although the number of the product, for example, from here we can see like it will need a complete RNA sequence, and then it will be translated into protein level. And because of we have more gene right here, so which means we have a higher a longer length of the gene right here, which means like more read can be aligned into the gene. So the gene length should be considered. We can divide it by the gene length for more directive comparison. And then the third issue is about the outlier. For example, if we have many genes in compare between sample A and sample B, and then we decide to minimize the issue of the total read. However, in the sample A we have a very special gene, is the outlier. It creates a lot of gene, and if we divide it by the total read count for all of the gene, so it might skew your data, which means like for example, although we can see right here gene X is on sample A is a higher expressed than gene X in the sample B, but actually if we divide it like that, then gene X on the sample A will be lower expression than gene X in the sample P. And because of that we need to have like many methods to normalize the data before we compare between them. So overall is the basic idea about RNA-seq, and then after understanding that we can use it to use algorithm, the available tools to analyze.